Hello students, welcome to EPG Pathshala. I am Dr. M.N. Gupta from IIT Delhi, and today we are going to talk on module Autoimmune Diseases from paper Immunology. We have seen how overreaction by immune system results in many hypersensitivity reactions. In all cases, this overreaction we saw results in damage to host tissues. Somewhat similar consequences result when immune system starts attacking self-antigens. Some aspects of it are already covered when we discussed tolerance. So it will help to recollect the discussion. Also we saw in type 5 hypersensitivity reactions which are caused by chronic stimulation that this type of hypersensitivity overlaps autoimmune phenomenon. So the objectives of this module are to look at the spectrum of autoimmune diseases and their prevalence, to understand factors which are responsible for autoimmune diseases, to understand various mechanisms of autoimmunity, and to briefly look at pathogenesis and therapy of autoimmune diseases. So the concepts involved are autoimmunity, what are autoimmune diseases, how these could be organ specific or non-organ specific, and how the various parameters decide or define the contours of autoimmunity and autoimmunity diseases, namely age, gender, genetic factors, infections happening, and the nature of the autoantigens. Autoimmunity is mediated by acquired immunity mechanisms which starts operating against self-antigens. In some cases, the disease is due to primarily an autoantibody or T cells or by both. Whatever are the primary causes, T cells play an important role, which is not surprising as these cells are involved in regulation of immunity. Again, the irony is that no new molecules or cells are involved. It is just the breakdown of the balance among various factors which are involved anyway in the usual immune processes. The autoimmune diseases in organ-specific disease involve attack on specific self-antigens. Different organs, multiple tissue sites in many cases share these self-antigens that is apparently responsible for systemic autoimmunity. Systemic lupus erythematosus is an example where widely distributed antigen is involved. Anti-DNA antibody is produced in this disease. A given individual can develop autoimmune disease of more than one type. Thyroid autoimmunity can be associated with gastric autoimmunity. Celiac disease can be associated with Sjogren syndrome, type 1 diabetes, and thyroiditis. We had referred to Hashimoto's thyroiditis while discussing Graves' disease as an example of type 5 hypersensitivity. Hashimoto's disease is in fact one of the early examples where an organ-specific autoimmunity was identified. The disease is most common in middle-aged women and leads to promotion of goiter, promotion of the formation of a goiter due to hyperthyroidism. The thyroid gland gets infiltrated with mononuclear phagocytes and lymphocytes. The gland in this disease often shows regeneration of thyroid follicles, which distinguishes it from primary myxedema, in which thyroid shrinks and is nearly destroyed. Hashimoto's thyroiditis and SLE mentioned above are the extreme ends of the list of organ-specific and systematic autoimmunity. This slide shows the entire spectrum of the autoimmune diseases. On the top are the organ-specific diseases. As we go down the list, gradually the list morphs into the autoimmune diseases, which are non-organ specific. Naturally, there are remarkable overlaps at each end of this spectrum. Thyroid antibodies occur quite often in pernicious anemia patients who also generally have stomach autoimmunity. We mentioned this earlier. Similarly, stomach autoantibodies are often found in patients with thyroid autoimmunity. Among systemic autoimmunity, clinical picture of SLE is often associated with features of rheumatoid arthritis. However, overlap of diseases at either end is very rare. Although in non-organ specific diseases, symptoms appear in skin, joints, kidney and muscle, specific organs are more markedly affected. It may seem that the classification into organ specific and systemic may not be strictly valid. Its validity originates in the fact that within an individual or family, either organ specific or systemic autoimmunity diseases cluster together. Look at from another point of view. The organ specific antibodies, say in thyroid and stomach, may occur together in an individual, but then that individual is highly unlikely to have non-organ specific antibodies. It is estimated that nearly 3.5% of the population have autoimmune diseases. The most common are Graves' disease, type 1 diabetes, pernicious anemia, rheumatoid arthritis, thyroiditis, CD, vitiligo, MS and SLE. Among themselves, these together account for 94% of all cases. Other autoimmune diseases together represent only 6% of the incidences. Women are estimated to be affected 2 to 7 times more, but this number can vary and is as high as 10 times for SLE. Hashimoto's thyroiditis and SLE mentioned above are the extreme ends of the list of organ-specific and systematic autoimmunity. This slide shows the entire spectrum of the autoimmune diseases. On the top are the organ-specific diseases. As we go down the list, gradually the list morphs into the autoimmune diseases, which are non-organ specific. Naturally, there are remarkable overlaps at each end of this spectrum. Thyroid antibodies occur quite often in pernicious anemia patients who also generally have stomach autoimmunity. We mentioned this earlier. Similarly, stomach autoantibodies are often found in patients with thyroid autoimmunity. Among systemic autoimmunity, clinical picture of SLE is often associated with features of rheumatoid arthritis. However, overlap of diseases at either end is very rare. Although in non-organ specific diseases, Symptoms appear in skin, joints, kidney and muscle. Specific organs are more markedly affected. It may seem that the classification into organ specific and systemic may not be strictly valid. Its validity originates in the fact that within an individual or family, either organ specific or systemic autoimmunity diseases cluster together. Look at from another point of view. The organ specific antibodies, say in thyroid and stomach, may occur together in an individual, but then that individual is highly unlikely to have non-organ specific antibodies. It is estimated that nearly 3.5% of the population have autoimmune diseases. The most common are Graves' disease, type 1 diabetes, pernicious anemia, rheumatoid arthritis, thyroiditis, CD, vitiligo, MS and SLE. Among themselves, these together account for 94% of all cases. Other autoimmune diseases together represent only 6% of the incidences. Women are estimated to be affected 2 to 7 times more, but this number can vary and is as high as 10 times for SLE. The top half of this slide shows the factors predisposing the development of immunity. Some autoimmune diseases showing HLA associations are listed in the lower half. The role of gender has already been mentioned. Genetics is definitely important. Within a family, there is high predisposition to development of autoantibodies against the same organ-specific diseases. So, genetics control which organ is likely to be affected. MHC, which represents antigen or autoantigens, have an important role in this. There is a well-established correlation between HLA type and relative risk of various autoimmune diseases. While the percentage risk is more strongly correlated with MHC class II alleles, but in some cases strong correlation also exists with particular MHC class I alleles. The strongest correlation between HLA allele and percentage risk has been observed in ankylosing spondylitis with an individual with HLA-B27 is about 90 times more likely to develop the disease as compared to the one which lacks HLA-B27. Population studies show association of susceptibility to the IDDM with HLA genotype. IDDM stands for insulin dependent diabetes mellitus patients. Patients suffering from this invariably express HLA-DR1, HLA-DR3 and or HLA-DR4. These alleles are highly linked to HLA-DQ alleles which show high association with IDDM but is not detectable by serotyping. HLA-DR2 in fact seems to protect development of IDDM. It is rarely detected in IDDM patients. X represents here any other allele. Serotyping and trafficking and tracking family histories have been two approaches in looking at the genetic factors. Now, HLA genotyping has led to establishing more precise associations. This slide shows how the amino acid changes in the sequence of an MHC class II protein correlate with susceptibility to as well as protection from diabetes. The usefulness of HLA genotyping and the insights it can provide is actually best illustrated in case of IDDM. DR3 and DR4 alleles are tightly linked genetically to DQ beta alleles. Polymorphism at a specific position in DQ beta amino acid sequence has been found to be closely associated with the incidence. In the DQ beta sequence, aspartate 57 forms a salt bridge with arginine at the end of the peptide binding cleft. The diabetic patients instead have valine, serine or alanine at this 57 position. This abolishes the salt bridge and changes the stability of DQ molecule. This is a critical feature as similar molecular picture exists in case of non-obese diabetic mice which have serine after 57 position in the homologous chain. The correlation has been confirmed with transgenic non-obese diabetic mice which have aspartate 57 and show drastic reduction in incidence of IDDM. As in all autoimmune diseases, MSC genotype alone does not completely decide risk factors for the development of the disease. We have referred to serology as one tool in the study of autoimmunity. Before going any further, it is necessary to clarify that presence of autoantibodies is not a sure clinical diagnosis of a particular disease. Many decades back, Ernst Vidya-mitra postulated the autoantibodies being used to correlate with the disease should be ones which are present in all cases of the disease. It should be verified experimentally that the corresponding antigen, if used as an immunogen, causes the manifestation of the disease. The disease thus experimentally induced must show immunopathological lesions which are characteristic of the disease in confirmed patients. The disease should be transferable from one infected animal to a normal one by serum or lymphoid cells. These postulates were very helpful in that early era in bringing clarity to the studies on autoimmunity. To sum up the important caution, the finding of autoantibodies does not confirm, the mere finding of the autoantibodies does not confirm the corresponding disease. In any case, it should be clear from the earlier discussion that even in organ-specific diseases, one may find several autoantibodies in the serum. There are three possibilities when autoantibodies are detected. The obvious one is to suspect that autoantibodies are responsible for the observed lesions. It is also possible that a disease caused tissue damage, which in turn led to the development of the autoantibodies. For example, cardiac antibodies may develop after myocardial infarction. However, autoantigens released by simple trauma rarely leads to corresponding autoantibodies. Thyroglobulin injected animals produce thyroid autoantibodies and show thyroiditis, which is very similar to Hashimoto's disease in humans. We have been discussing genetic factors. This is also shown by the fact that not all strains of an animal respond similarly when used for inducing autoimmunity. Let us now return to the other factors which we mentioned, which are part of multifactorial dependence of autoimmunity. What is more complicated is that these factors do not influence the percentage risk independently. Genetics and gender, for example, interact as factors. Older humans and other animals are more prone. This is understandable since aging immune system is known to display laxity in immunoregulation. While in general females are more prone to autoimmune diseases, ankylosing spondylitis is almost exclusive to males. Neuroendocrine system is implicated. In a strain of female mice, removal of estrogen secreting gland, ovaries, or treatment with male hormone testosterone prevented SLE development. On the other hand, castrated males become prone to SLE. Correlation between infectious agents and autoimmune diseases have been known. Chronic infections with Borrelia burgdorferi is responsible for Lyme arthritis. This is transmitted by ticks to deer and rodents to people. Cross-reactivity with microbial antigens is known to also induce autoimmunity through infection. Trypanosome cruzi causes Chagas disease in humans as its antigens cross-reacts with antigens on neurons and cardiac muscles. An epitope common to heart A. Streptococci is responsible for rheumatic heart disease. In fact, once infection is controlled, the disease also goes away. Viruses are also known to induce autoimmunity. Viruses infecting lymphoid tissues are especially important in this regard as immunological control mechanisms are affected. SLE is possibly associated with the virus. It is also believed that juvenile type 1 diabetes may be due to viral infection. The antigens involved may be a cell surface, cytoplasmic, nuclear, or an extranuclear one. Generally, these are highly conserved proteins like heat shock proteins. A microbial infection elicits an anti-HSP response followed by response to the microbial components. Given the strong homology between microbial HSP and the human HSP cross-react. The autoantigens covers a wide range. Proteins, lipids, nucleic acids, and their conjugates all could act as antigens. Receptors, structural proteins, and enzymes all can act as autoantigens. In this slide, the nature of the autoantigens is mentioned along with the corresponding disease. Many enzymes have been identified as autoantigens in specific diseases. In celiac disease, autoantibodies have been detected against tissue transglutaminase, IgG antibodies to glutamic decarboxylase. In diabetes and thyroid peroxidase in thyroiditis are other well-known examples. Some more examples are shown in this list. Let us look at some more factors which are involved in the development of autoimmunity diseases. There are reports that the anti-nuclear antibody appear in the blood during treatment of ventricular arrhythmias with procainamide, and some small percentage develop SLE-like symptoms. Discontinuation of the drug at an early stage leads to disappearance of these symptoms. So in principle, drugs can initiate autoimmunity by unknown mechanisms. Thrombocytopenia, which is characterized by low platelet count, and anemia, which, which of course is involves the low count of the red blood cells. Both are often drug induced than it is realized. Immune deficiency is also associated with autoimmunity. This may seem like a paradox, but in some cases, immune deficiency leads to persistence of infection or inflammation. Deficiency of complement compounds C2, C4, C5 or C8 correlates with autoimmune diseases, particularly SLE. In such cases, immune complexes which are no longer efficiently cleared by the complement system accumulate. IgA deficiency also increases the risk of common variable immunodeficiency, which is called CVID. Then there are environmental factors. There is a disease called Goodpasture's disease, in which autoantibodies against type IV collagen are produced. In this, only patients who smoked had pulmonary hemorrhage. Injury to endothelial lining is necessary to expose basement membranes. Serum from patients with Wegener's granulomatosis contains antibodies which are reactive with neutrophil cytoplasmic granules. This Wegener's granulomatosis has necrotizing vasculitis due to antibodies against a neutrophil protease called proteinase 3. An infection results in cytokine activated neutrophils translocating the antigen to the cell surface. This results in severe vasculitis. This slide shows how T cells sometimes help for intermolecular complexes in the induction of autoimmunity. In normal individual, tolerance is an inherent part of immune system which avoids autoimmunity. Hence, self-reactive lymphocytes are either eliminated or fail to produce enough antibodies or are unable to present these antibodies in enough concentration. Release of a sequestered antigen can be followed by its uptake by an APC, which now presents it to TH. In another route, a cell expresses MHC class II, which it should not, and thus becomes an antigen presentation cells for an autoantigen. Yet, third mechanism of autoimmunity, TS and other cells are unable to send adequate suppression signals to TH, which becomes active. Lastly, polyclonal antigens bypass TH control. T cell involvement is critical to development of autoimmunity. Patients with thyrotoxicosis have thyroid specific T cell clones. Anti-CD4 helps in RA. The role of MHC in autoimmunity is due to their role in inappropriate presentation to T cells. In systemic autoimmunity, often anti-antigens involved are not identified. It is possible that T cells recognize idiotypes. Autoantibodies produced at the low level from immune complexes with autoantigens. These complexes are taken up by APC, which include B cells. These APC process the complexes and prevent antibody idiotype to Th cells. The idiotype specific T cells help corresponding B cells. We have earlier referred to an example of molecular mimicry where rheumatic heart disease is due to identity of epitopes between group A streptococci and heart muscle. In this case, these specific B cells receive stimulatory signal from streptococci epitope specific Th cells. This slide illustrates this T cell bypass mechanisms. The plasma cells secrete antibodies which cross-react with heart muscle cells. Infection by Epstein-Barr virus in humans lead to polyclonal activation of B cells of all specificities, not completely eliminated by central tolerance mechanisms. These of course include those against self-antigens. Patients with mononucleosis, which is caused by this virus, produce IgM against several self-antigens including DNA. Class switching does not occur, indicating that Th cells are not involved. Control of infection leads to disappearance of these IgM antibodies. Regulating T cells, TREGs, maintain self-tolerance. Decrease in TREGs or impairment of their function has been identified as contributory causes in the several autoimmune diseases in humans. TREGs keep autoreactive Th under control through several ways, which include antigen-specific suppression. Cytokines like IL-10, T4-F-beta play a role in suppression. Either over-responsive Th cells or impairment of the Th cell functions tilt the balance towards autoimmunity. We have earlier pointed out close relationship between hypersensitivity and autoimmunity. In autoimmunity, the tissue damage ultimately operates via one of the hypersensitivity reaction. Hemolytic anemia results from the antibodies directed against RBC. Thrombocytopenia results from antibodies directed against platelets. These are type 2 hypersensitivity mediated autoimmune reactions. SLE produces a wide variety of autoantibodies. Anti-DNA are the most significant among these. The immune complexes deposit in glomeruli. Similarly, RA has immune complexes deposited in joints. The tissue damage follows hypersensitivity type 4 mechanisms. Finally, Graves' disease and myasthenia gravis involve chronic stimulation of TSH. Tissue damage follows type 5 hypersensitivity reactions. Finally, this slide shows the possible therapy of autoimmune diseases. The treatment approaches can be a replacement therapy in which the targeted autoantigen is replaced. For example, thyroid hormone for thyroid autoimmune disease, insulin for type 2 IDDM. Suppression of autoimmune process is another approach. In this case, non-steroid anti-inflammation drugs and glucocorticoids can reduce inflammation. Increasingly, monoclonal antibodies are emerging as the most important therapy in case of many autoimmune diseases. There are some other approaches which are at the clinical trial phase. For example, antigens given via the oral route to re-establish tolerance. To summarize the lecture, we learnt about autoimmune diseases can be organ-specific or non-organ specific. Age, gender, genetic factors, infection also influence autoimmunity. Many mechanisms of autoimmunity are known. T cells play a crucial role. Tissue damage follow hypersensitivity mechanisms. We also learned something about replacement therapy and suppression of tissue damage which follow hypersensitivity mechanisms. Replacement therapy and suppression of autoimmunity can be tried to minimize damage. Tolerance, hypersensitivity reactions, and autoimmune diseases are all interlinked. Autoimmunity is a failure of tolerance mechanisms. The tissue damage is due to the mechanism which we discussed while looking at various hypersensitivity reactions. The diagnosis of autoimmunity is not always straightforward. Here, detection of an antibody does not mean that the corresponding disease will manifest itself. It is necessary to remember this so that we do not have a false alarm. There is a lot which we do not yet understand about the interplay of various factors which predisposes one towards autoimmunity. Thank you.